Good morning. I'm going to present an encoding model of temporal processing in human visual cortex, and this is work done with Anthony Stigliani, who recently graduated from my lab. So, two seminal papers, one by Carl Friston in 1994 and another by Boynton and Heeger in 1996, provides a mathematical foundation for analyzing fMRI data. And basically, they showed that the relationship between the stimulus and the bulb is roughly linear, and using a linear systems approach, one can predict the bulb response from any stimulus by convolving the stimulus with the HRF, or hemodynamic response function. And this is what's also referred to as a GLM, or the general linear model, which is commonly used to analyze fMRI data. But as John mentioned, and as also observed from earliest fMRI days, the linearity fails in many situations. One situation this fails is when stimuli are presented for short durations. For example, the prediction of the linear model doesn't follow the measured data from V1, as shown by Byrne and colleagues already in 2001. There are additional nonlinearities, especially in high-level visual areas. And these actually nonlinearities that are seen when the stimulus is presented for a prolonged duration. So Gilai Dotan and colleagues showed a single image for 21 seconds, and they observed reduced responses in high-level visual areas or adaptation for the prolonged stimuli. So what are the sources of the nonlinearities? Let's look at the black box of the brain. So there are two components to the black box of the brain. One component is a neural response that is evoked by the stimulus, and another component is a hemodynamic response. So nonlinearities may arise either from a neural origin or from a hemodynamic origin. So the first question that we ask is, what is the source of the nonlinearities, neural or hemodynamic? So to do so, we designed an experiment that was predicted to induce substantial nonlinearities. So subjects participated in 3 different experiments. In all the experiments, they saw phase-scrambled images, and they did a central fixation task. In the first experiment, in each trial, we showed subjects a single image for the entire duration of the trials, and trials varied in duration from 2 to 30 seconds. In the second experiment, subjects saw in every trial 30 different images. Each image was shown very briefly for 33 milliseconds, and we varied across trials the interstimulus interval. In the third experiment, subjects also saw in each trial 30 different images, but images were shown continuously, and image duration varied from 67 milliseconds to 1 second. And critically, across all 3 experiments, we used exactly the same trial duration, which was 2, 4, 8, 15, or 30 seconds. So first, let's look at what does the GLM predict that V1 responses should look like. So according to the GLM, experiment 1 and experiment 3 should produce exactly the same responses because the total duration of stimulation is identical. Furthermore, the GLM predicts that responses will increase from 2 to 4 to 8-second trials, and the response amplitude would plateau thereafter. The GLM also predicts that responses in experiment 2, when we have these briefly presented images, is going to be substantially lower, and the reason is that the images are shown for only a small fraction of the trial, ranging from half of the trial to 1/30 of the trial in the 30-second blocks. So how does V1 data look compared to the prediction of the model? If we look at experiment 1 data, it actually follows the prediction quite nicely, that responses increase from 2 to 4 to 8-second trials and then plateaus thereafter. However, the linear model vastly underpredicts responses in experiment 2, which are surprisingly high, especially given that the simulation is a fraction of the duration of the trials. And in fact, having briefly presented images for less time can produce stronger responses in the shortened trial than a single image. Responses in experiment 3 also deviate from the prediction of the linear model because responses are higher than both experiments 1 and experiment 2. And this suggests that both the duration and the number of the stimuli in a trial affect V1 responses. So the question is, can modeling neural responses in millisecond resolution predict the observed V1 responses? So to model the V1 responses, we leaned on a large neuroscientific literature that suggests that in early visual cortex, there might be 2 temporal channels. One channel is a sustained channel, and this can be modeled with a neural impulse response function, or IRF, and it has a monophasic IRF that's shown here in blue. The second channel is a transient channel, and it is characterized by a biphasic impulse response function that functions very much like a derivative, so it codes changes in visual stimuli. However, when the stimulus is longer than the duration of the IRF, which is pretty short, it's 100 milliseconds, it predicts no neural response. So to implement the two-temporal channel encoding model, and a version of this model was suggested initially by Horiguchi and colleagues, what we did is instead of going directly from the stimulus to the bold response, what we're going to do is we're going to take the stimulus, predict the neural response in each channel, and then use the neural response to predict the bold response. So basically, we code the stimulus in millisecond resolution, we convolve it with a neural IRF, and we could get the predicted response in the sustained channel. We do the same for the transient channel, and in the transient channel, we added a nonlinearity in the form of a quadratic nonlinearity, which predicts increased neural response both for stimulus onsets and offsets. To predict the bold response, we're still going to assume linearity of the bold response, so we're going to convolve it with an HRF, and then we're going to use a general linear model to estimate the contribution of each channel, and we're going to sum this up to get the bold response. And the thing that you should notice is that what's going to happen is like fine changes in time in the neural signal are going to translate to amplitude effects in the bold signal. So how does the model do? So the first thing that we did, we took the data from experiments 1 and 2 and see if our model can predict the V1 responses. So we fit our model, and this is the estimated contribution of the sustained channel for each experiment shown in blue. This is the contribution of the transient channel shown in red, and the sum response across both channels shown here in black really quite nicely follows the bold response. Of course, the real test of the model needs to be testing how well the model can predict left-out data. So we're going to take the same model fits that we got from experiment 1 and 2 and see if we could use that data to predict experiment 3 data. So this is the contribution of the sustained channel in experiment 3. This is the contribution of the transient channel in experiment 3. And again, the sum response fits quite nicely our data. So overall, 3 experiments, our model predicts between 50% to 70% of the variance of our data. However, you could ask maybe other models, maybe hemodynamic models, could also explain our data. So we use the same kind of approach and tested how well other models can predict our data. In particular, we look at 3 types of hemodynamic models, the GLM, a hemodynamic temporal derivative model, and the balloon model that is supposed to account for nonlinearities in the bold response. And you can see that the hemodynamic models do a good job of predicting experiment 1 and 3, but do very poorly at predicting experiment 2 data. Basically, they really vastly underestimate the stimulus. We also read John's papers, and we implemented 3 versions of his compressive temporal summation. And the CTS model does much better than hemodynamic model, suggesting that modeling the neural responses is better, and especially it captures the strong responses in experiment 2. However, our model does a better job than the CTS model in predicting responses in experiment 3, where we vary both stimulus duration and number. So the results of the first set of experiments suggest that neural nonlinearities can account for the observed bold nonlinearities, and in particular, a 2-channel temporal model can predict V1 responses to stimuli that vary in time all the way from 33 milliseconds to 30 seconds. The question is if we can implement these kind of models to learn something new about temporal characteristics of the visual system beyond V1. And we're interested in looking at temporal characteristics of regions across 2 processing streams. The first processing stream is the so-called ventral stream, is a stream that starts in V1 and ascends towards ventral temporal cortex, and it is involved in object recognition. Another stream is a stream called the lateral stream. It ascends from V1 to the superior temporal cortex, and this is a stream that is involved in processing the dynamic aspects of visual stimuli. Additionally, as you can infer from this image, both streams contain face selective regions shown here in red and body part selective regions shown here in green. So it also lets us inquire whether there are different temporal characteristics across regions processing different domains. So we examine multiple hypotheses of how temporal characteristics can vary across the visual system. So one possibility is that the lateral stream that's involved in dynamic processing might be dominated by the transient channel, but the ventral stream that's involved in object recognition might be dominated by the sustained channel. Another possibility that we examined is whether there might be temporal differences across domain. Perhaps body parts are more dynamic than faces, so the body part selective regions independent of stream are going to be dominated by transients, but the face selective regions might be dominated by sustained responses. A certain alternative that's not mutually exclusive is that there might be differences across early and later visual areas. And indeed, there's a large literature that suggests that higher order areas have larger and later temporal integration windows compared to earlier regions. So to measure responses in high-level visual areas, we had to make some changes to our experiments. Specifically, the first change that we did is we showed stimuli that drive these high-level visual areas in the form of bodies, faces, and words. In addition, we made some slight changes to the design. In experiment 3, we put a short gap between stimuli, so we have the same number of transients in experiment 2 and 3. And third of all, we slightly changed the durations of the trials that are now 3, 5, 10, and 20 seconds. So first look at the responses in this experiment in a body part selective region and the ventral stream. You can see that this region behaves in a very different way than V1 in that responses do not accumulate with trial duration. In addition, when you show a single image for 20 seconds, you really see this adaptation happening throughout the trial. However, when you show 30 very briefly presented images, you can see that there's robust responses in this region. And furthermore, when you change both the number and the duration of stimuli, you get this highest response, again suggesting the additive effects of transient and sustained stimulation. We see a very different profile response in a high-level body part selective region and the lateral stream. In fact, this region only responds to the onsets and offsets of the stimulus, and basically there's very little response during the sustained visual presentation. However, when you show 30 very short images, you get really robust response in this region. And furthermore, when you show 30 images for even a longer duration, you don't get really any additive response, and this really suggests that the transients are driving responses in this region. So what we're going to do is now use our two-channel model to predict responses. However, we made some changes in this model to be applicable for high-level visual regions. The first thing that we did is that we used optimization techniques to infer the time window of integration of the neural IRFs in higher visual areas. And in addition, we added a nonlinearity in terms of an exponential to account for adaptation for continuous stimulus. In addition, in the transient channel, in addition to modeling the duration of the IRF, we changed the nonlinearity to a compressive nonlinearity in the form of two sigmoids, and we independently infer the parameters for the offset and onset responses. But afterwards, the model is the same. We're going to convolve each channel with the HRF and estimate the contribution of the different channels with the beta weights. So to test our theoretical predictions, one of the things that we're going to look at is how much each channel contributes to the response of each region. So if we look in V1 in this experiment, even though we change the stimuli, we still get contribution from both the sustained and transient channel. However, if we look at our lateral body part selective region, you can see that it's really just driven by the transient channel. There's no sustained response. But if you look at our body part selective region and a ventral stream, you get both sustained and transient contribution. And this profile was true for other regions in the lateral stream and other regions in the ventral stream. So basically, using these stimuli in this experiment, we're able to reveal significant differences in temporal processing across the ventral and lateral processing stream. We can also use the model parameters to examine the temporal properties of the neural IRFs in these streams. So looking at the sustained channel, we can look at the ventral stream. And one of the things that you can see is that the temporal integration window is later and more extended in the ventral stream areas compared to V1. And in addition, you can see that the adaptation increases from V1 to higher level visual areas on the ventral stream. If we look at the transient channel, we can see that also higher level regions in the lateral stream have a more delayed transient response compared to V1. And these regions actually show similar onset and offset nonlinearities, suggesting that they equally contribute to the signal. In the ventral streams, there's also a transient component, and you can see that this transient component is again later than V1. However, different than the lateral regions, we see an asymmetric compression between the offset and onset responses. And what really this means that there's much greater offset responses in these regions compared to onset responses. So using this approach, we're able to detect important differences in temporal processing across lateral and ventral temporal areas. And our data shows that the lateral regions are dominated by transient responses, and this likely means that they code the changeable aspect of the visual input. In the ventral stream, we observe both sustained and transient contribution, and we think that the transients are important for the ventral stream because the onset transient might code that there's a novel stimulus that you're looking at, and the offset contribution might give you some kind of memory information about the stimulus after it's no longer visible. So to conclude, I hope I've shown you that neural encoding models with millisecond resolution and neural nonlinearities can predict fMRI responses to a large range of temporal stimuli, ranging all the way from 33 milliseconds to 30 seconds. And critically, I feel that this approach is really important because it enables linking fMRI responses to the timescale of neural computation in Cortex. And I concur with John that I think this kind of approach can be used across the brain beyond the visual system. Okay, thank you for your attention.